Hey guys, Miss B here. So I'm going to walk you guys through the pre-lab practice from Thursday, how I would answer these questions. First off, don't forget to use your resources. I have this How Do I Identify the Variables and Groups resource posted on Google Classroom. It's under the quick reference material section, which is one of the first sections you see when you go to the classwork tab on My Google Classroom. Okay, so scenario 1, we have protease and pH. And remember, pH is a measure of how acidic or basic something is. Okay, and so it says you're tasked to discover in which pH protease functions best. Protease is an enzyme that breaks down proteins into chains of amino acids. That's bolded, so I'm also going to highlight it so that I remember it. Okay, you set up 3 different beakers. The first has a solution with pH 2, 10 milliliters of protease, and 10 milliliters of protein. The second has a solution with pH 7, 10 milliliters of protease, 10 milliliters of protein. The third has a solution with pH 13, 10 milliliters of protease, and 10 milliliters of protein. I think it would be best if I drew a picture of this so that we have a good idea of what this lab is. Okay, so beaker 1, how many? There's 1, 2, 3 beakers. Let's draw those first. Okay, first one has pH of 2. So this is pH of 2, and remember that is acidic. It has 10 milliliters of the enzyme, 10 milliliters of protein. Okay. The second has a solution with pH 7, which we remember that is neutral. It has the same amount of enzyme and substrate. So I'm just going to copy and paste this over here. Okay, sorry, that took a second. All right, and then the third one, sorry, I'm going too high up. It says pH 13, same amount of enzyme and substrate. Remember, pH 13 is basic. All right. Okay, so I want to find my independent variable, dependent variable, and controlled variables to start. So, what is different between each of these groups? That would be my independent variable. What am I changing? These are all the same amount of enzyme, same amount of substrate, but it looks like I have different pH levels. So we'll say pH level is my independent variable. Okay, so what am I measuring? I don't know if that's necessarily clear from the drawing, so let's go back to the reading. It says you're tasked to discover in which pH protease functions best. So how do I know if protease is functioning? The next sentence says protease is an enzyme that breaks down proteins into chains of amino acids. So I'm going to guess that protease is functioning if I see amino acids, because protease is our enzyme. Protein sounds like our substrate because it's what protease is working on. And then it sounds like amino acids. Those are our products because it says that the enzyme protease breaks down proteins into amino acids. Okay, so I'm going to guess that at the end of the lab, I'm going to measure how many amino acids there are. And if there's a lot of amino acids, that probably means protease did its job. So we'll say amount of amino acids. All right, our controlled variables, what are we not changing? Well, we already kind of pointed this out, right? pH is changing, but it looks like the amount of enzyme is staying the same and the amount of substrate is staying the same. Okay, so let's pick our control group out of these three options. We know that control group is usually like 0, right? But in the case of pH, there is no 0 pH. So we want to go with probably the most normal pH. And I would say that pH 7, the neutral pH, probably is our normal pH. So that must mean the other two, those are our experimental groups. So pH 2 and pH 13. All right, number 2 says write a hypothesis. What do you already know about how pH affects enzymes? Which pH do you think protease will work best in? Why do you think that? Write at least 4 sentences. All right, I'm going to zoom in here a bit. So what do I already know about how pH affects enzymes? I know that if an enzyme is not in the correct pH, it will lose its shape. And if we remember, the vocab word for that is denature. It will denature. So which pH do I think protease will work the best in? Well, pH 2 is super acidic. That sounds pretty harsh. pH 13 is pretty basic. That also sounds harsh. So I'm going to guess that protease will function best in the neutral solution, pH 7, because I don't think that will cause the enzyme to lose its shape. So I think protease will work best in pH 7 because it's the least harsh environment. I know that protease would lose its shape if it's in the wrong pH. I also know that pH 2 and pH 13 are very harsh environments. So it's always best to kind of over-explain yourself at the hypothesis stage. I want to know exactly what you're thinking, exactly why you're making that guess.